will I do? So again, outro. See. Sorry, nawawala yan ako internet. Nakisaro ko yan ako Zoe. Yan, kadinala to yan.
Okay, hindi na ka nagbabasa.
Okay. Oh yeah, so kadinala didi.
Okay, paano hawak yun?
Yan. Didi.
Okay.
Okay, good afternoon again. So before I start with our topic for this afternoon, it's about lipids or carbohydrates. I would just like to show you something para at least masuro sa unit akong pag-lecture ay or akong pagginakan. Huh? Okay. Amot makurit din rin nasa booted computer, may assistant. And guys, kindly my internet connection ko is unstable. Watch this.
Fats are an essential part of a healthy.
Can you hear it?
Yes.
Fats are an essential part of a healthy diet. They contribute to the taste and texture of foods, like the smoothness of guacamole and the flakiness of a croissant. Fats are also a major source of energy and a critical component of cells and tissues, and they also help absorb essential vitamins, and can be converted into other molecules like prostaglandins, which help cells communicate with each other. Fats have a 3-carbon backbone called glycerol, as well as fatty acid chains. The fatty acid chain is basically a string of carbon and hydrogen atoms. When an OH group from the glycerol molecule binds to a hydrogen from the fatty acid, an H2O or water molecule gets released, and the two molecules link up. If this happens once, the result is a monoglyceride. If it happens twice, it's a diglyceride, and 3 times makes a triglyceride. Now, there are various types of fatty acid chains, and one way to categorize them is by their length. In other words, how many carbons they have. Short-chain fatty acids have 2 to 5 carbons, medium-chain fatty acids have 6 to 12 carbons, and long-chain fatty acids have 13 or more carbons. Fatty acid chains are also categorized by the bonds connecting the carbons in the chain. A single bond is just one bond between the carbon atoms. And when a fatty acid chain has only single bonds, it's called a saturated fatty acid, because it has as many hydrogen atoms as possible, or it's saturated with them. Triglycerides with saturated fatty acids are nice and straight, so they pack together really well. And as a result, they're usually solid at room temperature. And the longer the saturated fatty acid chain, the more likely it'll be solid at room temperature. Carbons can also have double bonds between them though. And when a fatty acid has one or more double bonds, it's called an unsaturated fatty acid, because it's not saturated with hydrogen atoms. For every double bond, there are 2 fewer hydrogen atoms. Also, a double bond causes a kink in the molecule. They don't pack together as nicely as saturated fats. As a result, unsaturated fats are usually liquid at room temperature. Unsaturated fatty acids can be further classified, according to the number of their double bonds. Monounsaturated fatty acids are unsaturated fatty acids with just one double bond. Polyunsaturated fatty acids have 2 or more double bonds. Also, they can be classified according to their end, it's an omega-6, and if it's 9 carbons from the end, it's called omega-9. EPA, and docosahexaenoic acid, or DHA. EPA and DHA are marine sources of omega-3s. They're produced by microalgae and end up in the tissues of fish like anchovies, mackerel.
Nawala. Anay ha.
Salmon, and sardines. ALA is found in plants like flaxseed, walnuts, and canola and soybean oils. Our bodies can convert ALA into EPA and DHA, but it's an inefficient process that yields only small quantities. And that's why dietary recommendations include foods that have EPA and DHA. Omega-6 fatty acids are also usually polyunsaturated and include linoleic acid and arachidonic acid. Linoleic acid is found in oils like safflower, corn, and soybean oils. Arachidonic acid is found in animal sources like fish, meat, and eggs. Our bodies can convert linoleic acid into arachidonic acid, but once again the process is inefficient. Because ALA and linoleic acid can only be obtained in the diet, they're considered essential fatty acids. Omega-9 fatty acids are typically monounsaturated fatty acids, and an example would be oleic acid, and these can be made by the human body. Foods like canola and olive oil, as well as almonds, contain omega-9s. Now, looking at the double bond of this unsaturated fatty acid, like most unsaturated fats, it's got a cis configuration. In a cis configuration, the two functional groups are on the same side of the double bonded carbons. Now, when this happens, the fatty acid chain naturally bends. A molecule that bends does not pack tightly together, so it's a lot more fluid. Think about cooking oils, which are liquid at room temperature. Some fats are in a trans configuration, though. In a trans configuration, the functional groups are on opposite sides of the double bonded carbons, and this keeps the chain more straight and easier to pack. Trans fats result from a process called partial hydrogenation. In just plain old hydrogenation, hydrogens are added to cis fats to get rid of all of the double bonds. So let's say that you've got this triglyceride, and it has a total of 2 double bonds. So in this case, we'd add 4 hydrogens, 2 for each double bond. That turns the unsaturated fatty acids with cis double bonds into saturated fatty acids. Partial hydrogenation, on the other hand, refers to adding hydrogens to most, but not all double bonds. So let's say now that we add just 2 hydrogens. When this happens, some double bonds can be turned into single bonds by the hydrogens, but then they might reform. And what you end up with is an unsaturated fat, but some of those fats have trans double bonds. Partial hydrogenation is a process that happens naturally in the digestive tract of some animals, like cows and pigs, which is why trans fats can be found naturally in meat and dairy products. Trans fats are also created through the partial hydrogenation of liquid oils, a process that makes them solid. Partially hydrogenated oils have been largely removed from foods in North America and Europe, because trans fats have been associated with coronary heart disease. Although some foods might have more of one type of fat than another, the truth is that all foods are made up of a blend of fatty acids. When you eat a food like peanut butter, which has about 75% of its calories from fat, the body goes through a set of steps to digest and absorb the fatty acids. First of all, triglycerides are hydrophobic, so they form large globules of fat, like what you see when you pour oil in water. Enzymes called lipases in the saliva, stomach, and secreted by the pancreas can break down triglycerides into free fatty acids and monoglycerides. But working on the surface of a globule is inefficient. So to speed things up, bile salts produced by the liver break the large fat droplet into smaller droplets, which increases the surface area for the lipases to work. Once the triglycerides are broken down into monoglycerides and free fatty acids, these self-assemble into mixed micelles, which have a hydrophobic interior and a hydrophilic or water-loving exterior. The micelles glide through the watery environment of the intestinal lumen and reach the enterocytes in the intestinal wall. When they get to the enterocytes, the micelles release the fatty acids and monoglycerides, which diffuse into the enterocyte. Inside the enterocyte, the fatty acids and monoglycerides reassemble into triglycerides, and these get packed into a larger structure called a chylomicron. The chylomicron has lipids and proteins, so it's a lipoprotein. It has an outer membrane with phospholipids and proteins, and a hydrophobic core that has triglycerides, cholesterol, and fat-soluble vitamins A, D, E, and K. The chylomicron then leaves the enterocyte, but it's too large to get into the endothelial cells, so instead it enters a nearby lymphatic capillary called a lacteal. From there, the chylomicron floats in the lymph and flows into the thoracic duct, and then gets dumped into the blood, essentially bypassing the portal vein. Once in the blood, the chylomicron releases fatty acids and monoglycerides in peripheral tissues like muscle, which use them for energy, as well as adipose tissue which can store them. After delivering the triglycerides, the chylomicron shrinks in size and eventually gets engulfed by the liver. Now, fats play a super important role throughout the body. They have a huge number of health benefits, and those benefits can vary by the type of fat we eat. For example, polyunsaturated fats are precursors for hormone-like molecules called prostaglandins that stimulate endothelial cells that line blood vessels to release nitric oxide. Nitric oxide is a vasodilator, so that decreases resistance to blood flow and in turn lowers blood pressure. Polyunsaturated fatty acids can also help reduce the total and LDL cholesterol, and that's linked to lower rates of cardiovascular diseases like heart attacks and stroke. Long-chain omega-3 fatty acids like DHA and EPA can both help to lower plasma triglyceride levels, which also protects against cardiovascular disease. Finally, DHA is important in the development of eyes and brains of young infants. The impact of saturated fat on cardiovascular health is a little more complex. Generally speaking, it's recommended to keep consumption of saturated fat low, but just like with unsaturated fats, there are different types of saturated fats. Evidence suggests that different types of saturated fatty acids might have different effects on our cardiovascular health. Evidence also suggests that the health impact of reducing or replacing saturated fat in the diet depends on the nutrient that replaces it. For example, replacing saturated fat with polyunsaturated fat has been shown to benefit cardiovascular health, whereas replacing saturated fat with refined carbohydrate has not. Based on this, the National Academies of Medicine recommends consuming 20 to 35% of our daily calories as fat for a 2,000 daily calorie intake. That comes out to between 400 and 700 calories from fat, or about 44 to 78 grams. And it's not just about the amount of fat that we eat. The type also matters. The World Health Organization and the US Dietary Guidelines recommends that less than 10% of daily calories comes from saturated fats, and that trans fat consumption should be kept as low as possible. Alright, as a quick recap, fats are an essential part of our diet and health. Fatty acids are either saturated or unsaturated, and foods with fat always have a mixture of fatty acid types. Healthy diets emphasize mono and polyunsaturated fats over saturated fats, and trans fats are associated with an increased risk of cardiovascular disease. Improvements to our diet and health can be made by focusing more on the type of fat we eat and less on the amount.
Okay, thank you. Let's start. Lipids. So lipids are a heterogeneous group of compounds including fats, oils, steroids, waxes, and related compounds that are related more by their physical and by their chemical properties They are relatively insoluble in water and soluble in nonpolar solvents such as ether or chloroform. So it is an important dietary constituent, not only because of the high energy value of fats, but also because of essential fatty acids, fat-soluble vitamins, and other lipophilic micronutrients that are contained in the fat of natural foods. So the long-chain omega-3 fatty acids, dietary supplementation, the beneficial effects of these are chronic diseases including cardiovascular disease, Okay, prevention of chronic diseases, cardiovascular diseases, rheumatoid arthritis, and dementia. Adipose tissue is where your fat is stored. It also serves as a thermal insulator in the subcutaneous tissues and around certain organs. Nonpolar lipids act as electrical insulators, allowing rapid propagation of depolarization waves along myelinated nerves. Lipids transported in blood combined with proteins is in lipoprotein particles. So classifications of lipids are simple, complex, and derived lipids. The simple are fats and waxes. Fats are esters of fatty acids with glycerol, while waxes are esters of fatty acids with higher molecular weight monohydric alcohols. So esters of fatty acids is always contain an alcohol, and one or more fatty acids are complex classification of lipids. So phospholipids, glycolipids, and other complex lipids. In many phospholipids, the alcohol is glycerol, and it's called glycerophospholipids. But in sphingospholipids, it is sphingosine, which contains an amino acid, and lipoproteins may also be placed in that category. Derived lipids. They are formed from the hydrolysis of both simple and complex lipids. They include fatty acids, glycerol, steroids, other alcohols, fatty aldehydes, ketone bodies, hydrocarbons, lipid-soluble vitamins, and micronutrients and hormones. Some of these also act as precursor lipids in the formation of simple and complex lipids. Examples are your acylglycerols, your cholesterol esters, which are neutral lipids. So fatty acids are aliphatic carboxylic acids. They occur mainly as esters in natural fats and oils, and esterified form as free fatty acids and transport form in the plasma. Fatty acids that occur in natural fats usually contain an even number of carbon atoms. The chain may be saturated or unsaturated, as the video has presented. Saturated contains no double bonds, so that's why there are always hydrogen for each carbon atom. While unsaturated contains one or more double bonds, which frees hydrogen atoms. So a saturated fatty acid is an example, palmitic acid, 16 carbons. A monounsaturated fatty acid, oleic acid, has carbon at 1, 18 carbons at 1. A polyunsaturated fatty acid is a linoleic acid with carbon 18 at 2 double bonds. So fatty acids are named after corresponding hydrocarbons. The most frequently used systematic nomenclature names the fatty acid after the hydrocarbon with the same number and arrangement of carbon atoms, with OIC being substituted for the final E, the Genevan system. Saturated acids end in anoic. Octanoic is an example. Octa means 8, carbon 8, and unsaturated acids with double bonds end in enoic, like octadecanoic, decanoic acid or like acid carbon 18. So here at figure 21-2 is just a nomenclature for number and position of double bonds in unsaturated fatty acids. It has been also a bit discussed in the video. So carbon atoms are numbered from the carboxyl carbon number 1 on this side, and the carbon atoms adjacent to the carboxyl carbons number 2 and 3, 4 here are also known as the alpha, beta, and the gamma carbons, respectively. And the terminal methyl carbon is known as the omega carbon, which is on this side, or N carbon. So various conventions use delta for indicating the number and position of the double bonds. For example, delta 9, where your double bond is, indicates a double bond between carbons 9 and 10 of the fatty acid, and omega 9 indicated a double bond on the 9th carbon counting from the carbon, omega carbon. Saturated fatty acids contains no double bonds. So saturated fatty acids may be envisaged as based on acetic acid. It's the first member of the series in which CH2 is progressively added between the terminals CH3 and the COOH groups. Example of this is in table 21-1 shown in the book. Okay, so this one acetic, butyric, valeric, caproic, lauric, myristic, palmitic, and stearic, depending on the numbers of carbon atoms, 2, 4, 5, 6, and so on. Unsaturated fatty acids contain one or more double bonds. Again, monounsaturated and polyunsaturated eicosanoids are part of your unsaturated fatty acids. Your monosaturated are your monoethanoid, monoenoic acids, which has double band, one double band, and polyunsaturated, like your polyenoid, polyenoic, have two or more double bonds. While your eicosanoids, which is derived from eicosa, meaning 20 carbon, polyenoic fatty acids comprise the prostanoids, the leukotrienes, and the lipoxins. Prostanoids includes your prostaglandins, your prostacyclins, and your thromboxanes. In pharmacology, actually, you will also encounter this. So here is a table where unsaturated fatty acids of physiological and nutritional significance. Kindly just refer to your book at table 21-2. Okay. Prostaglandins. They exist in virtually every mammalian tissue, acting as local hormones. They have important physiologic and pharmacologic activities, and they are synthesized in vivo by cyclization of the center of the carbon chain of 20 carbons, eicosanoic, polyunsaturated fatty acids. Example of this is your arachidonic acid to form a cyclopentane ring. So found in the book also at figure 21-3 is prostaglandin E2. A related series of compounds, the thromboxanes, have the cyclopentane ring interrupted with an oxygen atom, or the oxane ring, found in your figure 24-4 in your book thromboxane A2, or your TXA2. Three different eicosanoic fatty gives rise to three groups of eicosanoids characterized by the number of double bonds in the side chains, which are examples are prostaglandin 1, 2, and 3. The different substituent groups attached to the rings gives rise to series of prostaglandins and thromboxanes, which are labeled A and B. Yeah, here. For example, the E type of prostaglandin, which has a keto group in position 9, whereas the F type is a hydroxyl group in this position. So I don't know where it is, but then again, the leukotrienes and lipoxins are a third group of eicosanoid derivative formed via the lipoxygenase pathway. They are characterized by the presence of 3 or 4 conjugated double bonds, and leukotrienes cause bronchoconstriction as well as being potent pro-inflammatory agents, which plays a big part in your asthma. What are the most naturally occurring unsaturated fatty acids, which have cis double bonds? The carbon chains of saturated fatty acids form a zigzag pattern, which extended at low temperatures. So at higher temperatures, some bonds rotate, causing chain shortening, which explains why biomembranes become thinner with increasing temperature. A type of geometric isomerism occurs in unsaturated fatty acids too, depending on the orientation of atoms or groups around the axis of double bonds, which do not allow rotation. If the acyl chains are on the same side, as it has been mentioned kanina, it will cause bending because it is on the same side. It's called a cis form. If on the opposite side it is trans, and in example is aniladic acid, the trans isomer of oleic acid. Double bonds in naturally occurring unsaturated long-chain fatty acids are nearly all in the cis configuration, the molecules being bent at about 120 degrees at double bond. Thus, oleic acid has a V shape, whereas aladic acid remains straight. Increasing the number of cis double bonds in the fatty acids leads to a variety of possible spatial configuration of the molecule. An example of this is arachidonic acid with 4 cis double bonds, so it is bent like a U shape. This has a profound significance for molecular packing in cell membranes and on the positions occupied by fatty acids in more complex molecules such as phospholipids. Trans fatty acids are present in certain foods, arising as a byproduct of the saturation of fatty acids during hydrogenation or hardening of natural oils in the manufacture of margarine. An additional small contribution comes from the ingestion of ruminant fat that contains trans fatty acids arising from the action microorganism in the rumen. Consumption of trans fatty acids is now known to be detrimental to health and is associated with increased risk of diseases, including your heart diseases and diabetes mellitus. It's led to improved technology to produce soft margarine low in trans fatty acids or containing none at all as of the moment. So what physical and physiologic properties of fatty acids reflects chain length and degree of unsaturation. That is why it's not healthy to eat saturated fat. The melting point of even-numbered carbon fatty acids increase with chain length and decrease according to unsaturation. A triacylglycerol containing three saturated fatty acids with 12 or more carbons is solid at body temperature, whereas if the fatty acid residues are polyunsaturated, it is liquid to below 0 degrees centigrade. In practice, natural acylglycerols contain a mixture of fatty acids tailored to suit the functional roles. Example of these are your membrane lipids, which must be fluid at all environmental temperatures and are more unsaturated than storage lipids. Lipids in tissues that are subject to cooling, for example, during hibernation or in the extremities of animal, are also more unsaturated. Omega-3 fatty acids. What are their anti-inflammatory and their health benefits? The long-chain omega-3 fatty acids such as linoleic acid, which are found in plants, the eicosapentaenoic acid (EPA) found in fish oil, and the docosahexaenoic (DHA) found in fish and algal oils. They have anti-inflammatory effects, perhaps due to their promotion of the synthesis of less inflammatory prostaglandins and leukotrienes as compared to omega-6 fatty acids. Their potential use as a therapy in severe chronic disease where inflammation is contributory cause is still under intensive investigation. Current evidence suggests that diets rich in omega-3 fatty acids are beneficial, particularly for cardiovascular disease and also for other chronic degenerative diseases such as cancer, rheumatoid arthritis, and Alzheimer's disease. Triacylglycerol, or triglycerides actually. According to the standardized terminology of the International Union of Pure and Applied Chemistry and the International Union of Biochemistry, the monoglycerides, diglycerides, and triglycerides should be designated as monoacylglycerols, diacylglycerols, and triacylglycerols, respectively. But still now, triglycerides is still being used. As the older terminology is still widely used, particularly in clinical medicine. So triacylglycerols are triglycerides. They are the main storage forms of fatty acid. The triacylglycerols are esters of the trihydric alcohol glycerol and fatty acid. There is mono and a diacylglycerol. That's why there are three, wherein one of two fatty acids are esterified with glycerol are also found in the tissues. These are of particular significance in the synthesis of the hydrolysis of triacylglycerols. So carbon 1 and 3 of glycerol are not identical. To number the carbon atoms of glycerol unambiguously, the stereochemical numbering system is used. It is important to realize that carbons 1 and 3 of glycerol are not identical when viewed in 3 dimensions. Enzymes readily distinguish between them and are nearly always specific for one or the other carbon. Phospholipids are the main lipid constituents of membranes, so plasma membranes, cellular membranes. Many phospholipids are derivatives of phosphatidic acid, in which the phosphate is esterified with one OH group of glycerol, and the other two OH groups are esterified to two long-chain fatty acids, or glycerophospholipids. Phosphatidic acid is important as an intermediate in the synthesis of triacylglycerols, as well as phosphoglycerols, but is not found in any great quantity in tissues. So this one, kindly just remember this, because we will come back to this every now and then. Okay, this is the phosphatidic acid, and this O actually The shaded phosphatidic acid is substituted for the substituents shown to form the phospholipids. So letter A, choline, it gimbal you, so it becomes your 3-phosphatidyl choline. B, ethanolamine naman na nagimbal igin butang DDHO. So it becomes your phosphatidyl ethanolamine. C, your serine being substituted here as a substituent, it becomes your phosphatidyl serine. And here, the myoinositol and so on, phosphatidyl glycerol. So this one, sphingolipids, as a sphingomyelin in which the phosphate is sterified to sphingosine, which is a complex amino alcohol, an important also component of your cell membrane. Both glycerophospholipids and sphingolipids have two long-chain hydrocarbon tails, which are important for their function in forming the lipid bilayer. This is the head and this is the tail. Okay, but in the former, both are fatty acid chains, while in this, there's a sphingosine tail and a fatty acid tail. Phosphatidylcholines or your lecithins and sphingomyelins are abundant in cell membranes. Your glycerophospholipids containing choline is most abundant phospholipids of the cell membrane and represent a large proportion of the body's store of choline. Choline is important in nervous transmission as acetylcholine and as a store of labile methyl groups. So this one. Dipalmethyl lecithin. It is a very effective surface active agent, important in your lung development. A major constituent of the surfactant, preventing adherence due to surface tension of the inner surfaces of the lungs. Absence from the lungs of premature infants causes respiratory distress syndrome. Most phospholipids have a saturated acyl radical in the SN1 position, but an unsaturated radical in the SN2 position of glycerol. Phosphatidyl ethanolamine or cephalin and phosphatidyl serine, which is found in most tissues, are also found in cell membranes and differ from phosphatidyl choline only in that ethanolamine or serine respectively. They replace the choline. So phosphatidyl serine also plays a role in apoptosis or the cell death, programmed cell death. This one, ethanolamine and your serine. Na-change sila didihan may phosphatidic acid. Sphingomyelins found in the outer leaflet of the cell membrane lipid bilayer and particularly abundant in specialized areas of the plasma membrane known as lipid rafts. They are found in large quantities in the myelin sheath that surrounds the nerve fibers. They are believed to play a role in cell signaling and in apoptosis. Phosphatidylinositol is a precursor of second messengers. The inositol present in phosphatidylinositol is the stereoisomer myoinositol. Phosphorylated phosphatidylinositols are minor components of cell membranes that play an important part in cell signaling and membrane trafficking. So this one, your letter D, ring. Phosphoinositides may have 1, 2, or 3 phosphate groups attached to the inositol ring. Phosphatidylinositol 4,5 biphosphate, so your PIP2, for example, is cleaved into diacylglycerol and inositol triphosphate upon stimulation by a suitable hormone agonist, and both of these act as internal signals or second messengers. Cardiolipin is a major lipid of mitochondrial membranes. Phosphatidic acid is a precursor of phosphatidylglycerol, which in turn gives rise to cardiolipin. This phospholipid is found only in mitochondria and is essential for the mitochondrial function of cells. Decreased cardiolipin levels or alteration in its structure or metabolism cause mitochondrial dysfunction in aging and in pathologic conditions, which includes your heart failure, hypothyroidism, and Barth syndrome or your cardioskeletal myopathy. So this one, the phosphatidylglycerol. Lysophospholipids are intermediates in the metabolism of phosphoglycerols or your lysolecithin. These are phosphoacyl glycerols containing only one acyl radical An example of this is your lysophosphatidylcholine or your lysolecithin, which is important in the metabolism and interconversion of phospholipids. This compound is also found in oxidized lipoproteins and has been implicated in some of their effects in promoting atherosclerosis. Plasmalogens occur in brain and muscle. These compounds constitute as much as 10 to 30% of the phospholipids of brain and also in the heart. Structurally, the plasmalogens resemble phosphatidylethanolamine but possess an ether link on the SN1 carbon instead of the ester link found in acyl glycerols. Typically, the alkyl radical is an unsaturated alcohol. In some instances, choline, serine, or inositol may be substituted for ethanolamine. The function of plasmalogen remains poorly understood, but it has been suggested that they may have a protective effect against reactive oxygen species or ROS. Glycolipids. Glycosphingolipids are important in nerve tissues and in the cell membrane. Glycolipids are lipids with an attached carbohydrate or carbohydrate chain. They are widely distributed in every tissue of the body, particularly in nerve tissues such as brain. They occur particularly in the outer leaflet of the plasma membrane where they contribute to cell surface carbohydrates which form the glycocalyx. The major glycolipids found in animal tissues are glycosphingolipids. They contain ceramide and one or more sugars. So galactosylceramide is a major glycosphingolipid of brain and other nervous tissue. They are found in relatively low amounts elsewhere. It's more of in the brain and nerves. It contains a number of characteristics like your C24 carbon 24 fatty acid, for example, the cerebronic acid. Galactosylceramide can be converted to sulfogalactosylceramide or your sulfatide, which has a sulfur group attached to the O in the 3 position of galactose and is present in high amounts in the myelin. Glucosylceramide resembles galactosylceramide, but the head group is glucose rather than galactose. It is the predominant simple glycosphingolipid of extraneural tissues, also occurring in the brain in small amounts. Gangliosides. Gangliosides are complex glycosphingolipids derived from glucosylceramide that contain in addition one or more molecules of a sialic acid. Uraminic acid is the principal sialic acid found in human tissues, and gangliosides are also present in nervous tissues in high concentration. They function in cell-to-cell recognition and communication and as receptors for hormones and bacterial toxins, which can be found in cholera toxins. So the simplest ganglioside found in tissues is GM3. So there's GM1. Okay, GM3, which contains ceramide, one molecule of glucose and one molecule of galactose and one molecule of neuraminic acid. G is ganglioside, M is a monocyano-containing species, and the subscript 3 is a number assigned on the basis of chromatographic migration. So GM1 is a more complex ganglioside derived from GM3. It is of considerable biologic interest as it is known to be the receptor in human intestine for cholera toxins. Other gangliosides can contain anywhere from 1 to 5 molecules of sialic acid, giving rise to your ditrisalongangliosides. Steroids play many physiologically important roles in humans. Although cholesterol is probably best known by most people for its association with atherosclerosis or heart disease, it has many essential roles in the body. It is the precursor of a large number of equally important steroids that include the bile acids, adrenocortical hormones, sex hormones, vitamin D, and cardiac glycosides. All steroids have a similar cyclic nucleus resembling phenanthrene rings A, B, and C, to which a cyclopentane ring D is attached. So let's just take a look at it na lang. So this one, your ABC rings with a D attached. It is important to realize that in structural formulas of steroids, a simple hexagonal ring denotes a completely saturated 6-carbon ring with all valences is satisfied by hydrogen bonds. unless shown otherwise. That is, it is not a benzene ring. All double bands are shown as such. Methyl side chains are shown as a single band and attached at the farther or on the methyl end, and this occurs typically at positions 10 and 13, constituting C atoms 19 and 18. A side chain at position 17 is usual, as in any cholesterol, and if the compound has one or more hydroxyl groups and no carbonyl or carboxyl groups, it is a sterol. Because of asymmetry in the steroid molecule, many stereoisomers are possible, but the chair form is the most naturally occurring steroids, okay, which is the more stable conformation of the steroid molecule. So this is the trans configuration, and this is the cis configuration of sterols. Okay. Cholesterol is a significant constituent of many tissues. Cholesterol is widely distributed in all cells of the body, but particularly in the nervous tissue. It is a major constituent of the plasma membrane and of plasma lipoproteins. It is found as cholesterol ester, where the hydroxyl group on position 3 is esterified with a long-chain fatty acids, and it occurs in animals, but not in plants or bacteria. Ergosterol is a precursor of vitamin D. Ergosterol occurs in plants and yeast and is important as a dietary source of vitamin D. When irradiated with ultraviolet light in the skin, ring B is open to form your vitamin D2 in a process similar to the one that forms vitamin D3 from 7-dehydrocholesterol in the skin. Polyprenoids. Polyprenoids share the same parent compound as cholesterol. They are not steroids but are related to them because they are synthesized from 5-carbon isoprene units. They include ubiquinone, which participates in the respiratory chain, in the mitochondria, and the long-chain alcohol, dolicol. They transfer carbohydrate residues to asparagine, residues of the polypeptide. The plant-derived polyprenoids include rubber, camphor, fat-soluble vitamins, your ADEK, ADEK, and your beta-carotene provitamin A. Lipid peroxidation is a source of free radicals. Peroxidation or autooxidation of lipids exposed to oxygen is responsible not only for deterioration of foods, but also for damages to tissues in vivo, where it may be a cause of cancer, inflammatory disease, atherosclerosis, and aging. The deleterious effects are considered to be caused by free radicals, molecules that have unpaired valence electrons, making them highly reactive. Free radicals containing oxygen examples are your ROO or your RO and OH. They are termed your ROS or your reactive oxygen species. These are produced during peroxide formation from fatty acids containing methylene interrupted double bonds. That is, those found in naturally occurring polyunsaturated fatty acids. Lipid peroxidation is a chain reaction in which free radicals formed in the ionization stage in turn generate more, and thus it has potentially devastating effect. So this one is the ionization and propagation and termination process for those ROS. Antioxidants. Antioxidants are used to control and reduce lipid peroxidation, both by humans in their activities and in nature. So propyl gallate, butylated hydroxy, anisole, and butylated hydroxytoluene are antioxidants which are used as food additives. Naturally occurring antioxidants include vitamin A, tocopherol, a lipid-soluble and compound, and urate and vitamin C, which are water-soluble. Your beta-carotene is an antioxidant at low O2. What are the two classes of antioxidants? You have your preventive antioxidants or your chain-breaking antioxidants. Your preventive antioxidants which reduces the rate of chain ionization, and your chain-breaking antioxidants interferes with chain propagation. Preventive antioxidants include catalase and other peroxidases such as glutathione peroxidase that reacts with ROOH, selenium, which is an essential component of glutathione peroxidase, and regulates its activity, and chelators of metal ions such as ethylene diamine tetraacetate and diethylene triamine pentaacetate, EDTA and DTPA. In vivo, the principal chain-breaking antioxidants are superoxide dismutase. This superoxide dismutase acts in the aqueous phase to trap superoxide free radicals, urate, and vitamin E, which acts in the lipid phase to trap ROO radicals. Peroxidation is also catalyzed in vivo by heme compounds, and lipoxygenases. They are found in platelets and leukocytes. Other products of autooxidation or enzyme oxidation physiologic significance include oxysterols and the prostaglandin-like isoprostanes. Formed from peroxidation polyunsaturated fatty acids such as arachidonic acid, they are used as reliable markers of oxidative stress in humans. Amphipathic lipids, self-oriented oil, water interfaces, they form membranes, they form micelles, they form liposomes and emulsions. In general, lipids are insoluble in water since they contain a predominance of nonpolar hydrocarbon groups, or your hydrophobic pole. However, fatty acids, phospholipids, ringolipids, bile salts, and to a lesser extent, cholesterol contain polar groups. Therefore, a part of the molecule is hydrophobic or water insoluble, and part is hydrophilic, which is water soluble. Such molecules are described as amphipathic. So these are just the examples of lipid membranes, micelles, lipid bilayer. This is the micelle, emulsion, oil in water, liposome, unilamellar liposome, and your multilamellar liposome. They become oriented at oil-water interfaces with the polar group in the water phase and the nonpolar group in the oil phase. A bilayer of such amphipathic lipids is the basic structure in biologic membranes. When a critical concentration of these lipids is present in aqueous medium, they form micelles. Liposomes may be formed by sonicating an amphipathic lipid in an aqueous medium. They consist of spheres of lipid bilayers that enclose part of the aqueous medium. Aggregation of bile salts into micelles and liposomes and the formation of mixed micelles with the products of fat digestion are important in facilitating absorption of lipids from the intestine. Liposomes are of potential clinical use, particularly when it is combined with tissue-specific antibodies as carriers of drugs in the circulation targeted to specific organs. They are used in cancer therapy. In addition, they are used for gene transfer into vascular cells and as carriers for topical and transdermal delivery of drugs and cosmetics. Emulsions are those much larger particles formed usually by nonpolar lipids in an aqueous medium. These are stabilized by emulsifying agents such as amphipathic lipids, phosphatidylcholine. These form part of a surface layer separating the main bulk of the nonpolar material from the aqueous phase. So in this phase, this is the hydrophilic and the hydrophobic part. In the aqueous phase, the hydrophilic, the hydrophobic part. So the head and the chain, bipolar. So, tapos. Thank you, thank you. May pakiana ka mo? Aday, boss. Added ako sinarigan.
Pwede na ka mag-talk.
Stop
Romel,
sharing.
Romel,
Sir,
Romel.
sir. Yes, sir.
Your carbohydrate is included in the second bimonthly.
Okay,
so
sir.
you make questions. Huh? I
Yes,
don't
sir.
know how many questions you can make. You just tell me how many. How much? How many questions you can have. So I will just add to make it 100.
Okay, sir.
So
Okay.
you can make 20 or 30 or whatever.
Yes, sir. Yes, sir.
This
When will
lecture.
I submit it to you, sir?
No, do not submit it to me. Submit it to Sasa.
Okay.
No, submit it to Sasa.
I'll make 20, sir.
Oh, sige, you just give it to Sasa, so that it will be
Encoded the canvas.
canvas. Yes.
Yes, sir. Yes, sir.
And then this lecture is not included. This will be for the second bi-monthly.
Okay.
Okay.
Yes,
All
sir.
right.
So, yan first buy pa man lang, ano, sir?
First buy.
Yes, sir. Yes, sir. So, it's for the second buy pa. With carbohydrates.
First week of February is the first buy.
Okay. So, di ribuli kini yan na, sir, na carbohydrate.
Really. It's not included. This will be in the second bi-monthly.
Doc, excuse me, Doc.
Yes.
Second by monthly na ada kita yan na, Doc.
Oh. I'm sorry. I am disoriented. Okay, second buy. This is included in the second buy. And no, second buy. This lecture will be for the third buy.
So, third
You
buy
mean
na.
You mean second sem, Doc?
Huh? For the second
Second
sem.
semester na. Second semester.
Second sem.
First by monthly of your
Okay.
second semester.
Our second buy will be in first week of February. I don't know the schedule yet.
May ambot ko may naging
Thursday ka na, Doc? Ano mo schedule, Doc, Biochem?
So yan na, sir, nga first buy o second buy, waray ako himoon lang.
Ang your this what you call this, the carbohydrate,
Carbohydrates.
carbohydrate, no lipid, just carbohydrate,
Okay, sir.
structure and function.
On the first lecture lang. Okay, sir. Okay, sir.
First lecture. So you can have as many questions as you can have. Just tell me how many questions you have made. So number it from 1 to whatever. 1 to 20, 1 to 30, 1 to 10. No? And just let me know. So I will just
Sabi
continue.
ko, sir, mas mapay 1 to 10.
Oh. It's up to you. You just tell me.
Yes, sir. Yes, sir. Thank you, sir.
So I can just go with the numbering. So if you ended up at 20, I'll start with 21.
Okay.
Okay?
Okay, sir.
All right.
Thank you, sir. Thank you so much.
Welcome. That's a good lecture.
You told me to read, sir. Sorry. Amo na ako nahimo.
Oh, that's a good presentation you have made. Yeah.
It's Mariel's presentation, sir, and Chloe's. Thank you so much.
They are more adept with this technology.
Yes, sir. Nagnasaot pagtikangan video kanina.
Yes, there's plenty of that at YouTube.
Yes, sir. I got it from there. Osmosis.
Yeah, that's a very big help to give you a bird's eye view right away.
Yes. Sure.
Okay, that's very good, Romel.
Thank you, sir. Salamat. Salamat. First year, salamat sa iyo, ha, mga classmate. Classmate kita Biochem.
Thank you, Doc.
Thank you,
Teacher
Doc.
ako kaya yung classmate kita Biochem. ha?
Thank you, Doc.
Bye bye.
Thank you, Doc.
Thank you. Sir, salamat. Thank you.
Thank you, thank you. Nag-out ako.